The analysis part of the software is subdivided in three main sections. A large screen displays all the data that is selected in the left part of the software. We have in the lower side a project data tree that allows to select different experiments that were run in this one job. These are represented as different folders shown here, and each of them have an identifier number shown here. When clicking on the plus sign of the folder, this will open up the experiment and show a representation of all the liquid periods that were executed in this experiment. We can see in this current example 8 different liquid periods were added. We can further see the name of the compound that was applied during this liquid period, and in the parentheses shown is the concentration applied. When opening an individual folder, it is possible to get an overview of all the sweeps that were run in this experiment. So in this particular liquid period, we have 16 sweeps. So we can directly navigate to an individual sweep if we intend to. The project data tree on the top side allows to select different levels of the data. We are currently started out by selecting the sweep plot, which shows us the raw data. The sweep result table will then show all the data that was extracted from this data, and after we continue with the analysis, we will further process the data to extract various parameters and analyze the data on different levels. This will be covered in more detail in another tutorial. In this section, it is also possible to set up filters, and they can be selected or applied on the different levels. A further possibility is to to look at the job and get a straight overview of the performance. It is possible to click on the Q plate, and here we see all the Q plates that were run in this job. In this particular job, 2 consecutive Q plates were run, and we can see an overview of the performance here. When clicking on the Q plate, we will see an overview of all the different wells with the identifier, position, and whether or not the priming was successful, the cell attachment was successful, seal formation and whole cell, and further parameters are shown on the right side. We have a graphical representation of the same in the lower part of the figure. Scrolling down in the table will give us a success rate overview of the particular steps, and this can be very useful for troubleshooting where the experiment went wrong. To adjust each of the figure for a presentation, for example, in the default setting we have a grid underlying the graph, and we also have shown in the red line here the applied voltage protocol, which can be useful to see really when a current was elicited at which voltage. If we do not want to show this for representation purposes, we can right click in the figure and say show grid, uncheck this one, and further click into the figure. Show protocol will remove the red line, the voltage protocol. Now we have the isolated current trace shown in this result. If we want to use this plot as a representative plot for a presentation, for example, we can right click again on the figure and export the drawing, or copy it to the clipboard, or save it in various files as shown here. It is further possible to adjust the different scaling on this plot. The y-axis, when right-clicking on the y-axis, we can choose to fix this axis span to a certain value that we choose. For example, as a maximum value, one could want to choose 400 picoampere. Now this will fix it for all the other figures as well. When jumping through the different data, we will always have 400 picoampere shown. Here we can also notice a different feature about the software. When selecting a certain sweep in a liquid period in an experiment, and we see this representation in the plot, we can simply click on another experiment and the exact same time point in this experiment will be selected.